What I want to do today is I want to talk a little bit about what to expect for class this week. So I want to talk a little bit about what to expect in lab. Some of you already had labs, so you know what to expect. Some don't have lab till Friday, so preview. We will talk about a little bit what to expect for lecture, especially because we are coming up on our second unit assessment. So this week we'll actually finish our unit 2, cell biology. And so I will talk a little bit about what to expect with the unit assessment, and then we can talk about muddiest points. So let's get into it. All right, so first, I just want to mention, so first, actually, what I'm going to do to kind of talk about this, sorry, I want to make sure everybody's getting in, is I'm actually going to share my screen here. So hopefully everybody sees Canvas. Just a reminder that our homepage kind of tells you exactly what to expect this week. And I tried to make it to where the assignments were equivalently spread. The most things I want you to note, so you can also look, of course, in the week 7 module, which contains all of our content. And the things I want to pay attention to primarily, is one, we do have another ePortfolio assignment. This one is regarding cell communication. So I saw several of you already started that. Good job, go ahead and post it. I think that originally this was going to be like in groups again, but actually I think that everybody's just posting in the discussion board. So when you peer review, just try to peer review just like 1 or 2 other people. Don't peer review everybody because then you would be peer reviewing the entire class. But do post your rough draft of this by Friday, or by Sunday, sorry. Kind of a fun thing. So I do want to mention that this week we are terminating cell bio, finishing our discussion of cell bio by talking about cell communication, which we are trying to, so we only have 3 lectures this week, 2 concept checks, and we have a lot of supportive content for cell communication. And that's because this topic tends to be one that is a difficult concept in biology in general, but one that is extremely important for everything. And so we wanted to make sure that you really got it. It's in essence, it's not challenging. It's just one of our very first concepts that's very abstract, right? So a little bit difficult to like observe. And so we wanted to just make sure you got a lot of supplement. So that's why we've got less lectures, but more supplemental. So the cell communication module should help. And then those lectures are good. I got to talk about plant communication, which surprisingly I enjoyed. So I don't normally love talking about plants, but I do like talking about how they can communicate. As well as that, so that's primarily for lectures. So as usual, lectures, ePortfolio assignment. I also want to mention that for lab, you will be working in groups just to submit your final drafts for your results and discussion. Order those so that they're in order. But also on the topic of cell communication, you have an individual assignment. And this is regarding more of like the lecture content, which is cell communication. So this is a simulated lab. It's again regarding cell communication. Make sure you read the background, make sure you read how to use this simulation tool. And then based on the information contained in these documents, there is a pre-lab quiz. And again, it just kind of covers material that's in these backgrounds and then a little bit about cell communication, kind of supportive material. And then like it's 10 questions, but 5 of them are just sort of like acknowledging that you understand the requirements of the simulation, because it can be a little bit finicky. So just pay attention to the requirements. And then after you take the or perform the simulation, there's like a very quick post-lab quiz, 4 questions. You'll notice that when you go through the simulation, you'll kind of be like participating in the lab, and it'll ask you a question about what you're doing or why it matters. The questions in the post-lab quiz are taken directly from the simulation. They're more general questions about cell communication, but they are questions that you will be asked during the simulation. So just pay attention to what you're doing throughout the simulation, why you're doing it, stuff like that. But all the questions and all the answers are found in the simulation. All right, so that's what we're doing for cell communication, a very important topic in biology, one that is inescapable. No matter what field you're going into, you're going to be talking about how that organism interacts with its environment, and therefore it's very important to understand how organisms interact with their environment. So that's what we're doing for lecture and lab. I also want to mention one thing. So I've got a couple of questions about the reading assignment. I just want to make sure that everybody understands that the reading assignment are for points, but the reading assignments, I have started opening them when class materials open. So this assignment opened up last Friday, and our next one will open up this coming Friday for the following week. I mention that because it's not due until the Sunday. So you like have over a week to complete this. And what I want to mention about that is that the reading assignments, you are able to, well, one, they're based on completion. So if you do it, you get full credit. But also these assignments are assignments that you can start and then like pause and come back to anytime. So I realized that they're very good assignments. Like the technology and the way it supports your learning is amazing. But I realized that like the time is a burden for those. It can be a burden. I try to put them in the recommended schedule when you have a little bit of less stuff going on, but what you can do is if you're finding that these assignments are a little bit difficult to complete in one sitting, that's okay. What you can do is you can do like, 10 to 15 minutes per day. It saves your work, come back to it based on completion. And so again, just, these are important. They do support your learning, but they're very low stakes, right? So treat them that way. I don't want them to be a burden, but I do want you to do them. All right, so that's kind of what we've got on the docket this week. Now, let's see. Oh, so this week also, if you go through the content, you're good, got the lecture concepts, you'll notice that our review for Unit 2, I can change this, is here. Unit 2 review, because I posted it. It does have to be proctored because of copyright purposes. I apologize. I know the technology is not ideal, but it does have to be proctored, and it is very similar to our last Unit 2 review in that you get 3 attempts at this assignment. Your best attempt will be the one recorded. So this time we did things a little bit differently because I believe in learning and improving, and that's what I want to do for this course as well, right? We're learning how to do stuff best as well. And so last time we had the Unit 1 review and Unit 1 checkpoint, we had that due like the Sunday that we finished the material. So you probably remember we had the macromolecules, and the Unit 1 review was like the same week that you learned macromolecules, and it was like you didn't have enough time to learn the content, even though you did great. But the purpose is learn, not to stress. So what we decided to do this week is we wanted to give you time to focus on the content that we're talking about this week before you jump into the unit assessment. So we have the Unit 2 review opening up on tomorrow. So it opens up, yeah, tomorrow. Oh yeah, it opens up, sorry, tomorrow at 12 o'clock a.m. So starting tomorrow at 12, like as soon as tomorrow starts, you could theoretically start doing your review. You have 3 attempts. I would recommend doing all 3 of these attempts since your best attempt is recorded and because you get more exposure to the potential questions. But these are not due until next Wednesday at 11:59 p.m. So you have from this Wednesday till next Wednesday to complete all 3 attempts of your review. Then the checkpoint for Unit 2 won't actually open until next Thursday at 12:01 a.m. and you'll have until the following Sunday to do it. So I'll have more information about that. But again, the purpose of that is that way we can wrap up Unit 2 content. We will have Tuesday of next week to do muddiest points from the whole Unit 2 content, whatever you want to ask about, and then you can take the checkpoint. So you could even ask questions on that, on your muddiest points about, you know, like review questions. Like, I got these questions wrong. I'm not really sure I understand this concept. So can we talk about it in the muddiest points? So you will have plenty of time to do the review, all 3 attempts, and you will have that before you start the checkpoint next week. We will have culminated the material. So everything is sort of hoping we can just sort of like, you know, obviously finish the material before you have to start knowing it for a test. So again, the checkpoint, hopefully everybody can see this. So the review opens this Wednesday, closes next Wednesday. You have 3 attempts. Best will be recorded. I do want to mention that it is the checkpoints, the assessments in this class are cumulative. So we want to make sure you're not forgetting the important material that we already did cover. So this time the review is 30 questions. This is 20 from what we've covered, so cell biology, Unit 2, which is weeks 5 through 7. And then there will be 10 questions included in the reviews from Unit 1, which was weeks 2 through 4, which was introduction to biology and chemistry, and of macromolecules and stuff like that. So that's your Unit 2 review. The Unit 2 checkpoint, which again will not open until next Thursday, it will actually be similar to the Unit 1 checkpoint. It will be worth 100 points. It will be 60 questions this time, so 50 questions from Unit 2, the cell bio, and then 10 questions from Unit 1. So way less questions from Unit 1, but they're still going to be there. And you will have an appropriate increase in time to take it. Unit 2 review attempts are not timed. You can also feel free to use resources. It's proctored, but you can access the textbook. You can get feedback. Unit 2 checkpoint is timed, and you can't access resources. So again, I'll push out kind of like written directives, but overall it will be like very similar to the setup for the Unit 1 review and checkpoint, kind of same rules apply. So I did see a question, so I'm gonna stop sharing for a minute. Allie?
Hi, yeah, I had a question about the Labster thing that we do this week.
Mm-hmm.
How will you be able to see that we did it? Does it just automatically submit somehow, or I was just wondering about that.
So that's a great question. So for the Labster for this week, it is based on completion. And so importantly, this is like very important, you have to make sure you have completed 100% of the simulation for it to go through. So if you see that you've submitted like 98% of the simulation, your score will not be recorded. So 100% of the simulation has to be completed. Yeah.
Okay, thank you. That makes sense.
Yeah. Rita?
Yeah, so I had a question. Did you
Yes.
6, like muddiest point in the PowerPoint? Because I don't see it in Canvas.
I'm having a little trouble understanding you.
Like
I think that your audio might have gone out. What I'm going to do is open, oopsies, what just happened? I'm going to open the chat. Rita, if you can't use your speaker, go ahead and use the chat. Oh, so okay, the question was the week 6 muddiest point PowerPoint. I haven't posted it yet. I just finished it right before class, but I'm recording this lecture. I will also post the week 6 muddiest points right after class today, so you'll have access to it. Yeah, I try to edit those right before class. So everything I want in there is included. Okay, so good questions. If you have more questions, again, raise your hand as we're going through this, or speak out. Okay, so there was a question about the organelle peer review. If you're having a hard time accessing some of them, just let me know, send me an email. Peer review the ones that you can. I know there was some issues with like the attached function on the discussion board for some reason. I'm not really sure what was going on there. But if you're having trouble, let me know. Peer review what you can, but if you can't access something, just send me an email and let me know. Okay, Curtis? Did you have a question? I can't hear you, but I see.
Sorry.
Oh, there you go.
Mike was on the wrong input.
That's okay.
To follow up the organelle peer review, is there like specific criteria that we need to address on there? Because I don't remember that being on the discussion board. I could be blind though.
No, you're good. So let me see. There was just essentially, if I go back here, like the kind of rubric for what you need to include in it. That's essentially just what we want to make sure that you include. So I think that was that in here? Yeah, it's just this creation should include.
Okay.
Yeah, good question.
All right, thank you.
Yeah. All right, so for feeling good.
Oh yeah, Dr. C.
Yeah.
I see on Sunday that for the osmosis results final draft that's posted twice. Is there a reason for that?
It's posted twice?
Yeah, just they're like the same assignment and this says they're both worth 20 points.
I'm trying to see. Are you looking in the modules?
I'm just on the dashboard looking at the Sunday part of
Okay.
my assignments.
So we had a little trouble with that because of the group settings. So when you submit it wherever you can. I contacted some groups, but submit it to this one, just the one that's in the modules. But we'll be able to find it wherever you submit it.
And just one last question. So is the lab, that's individual work this week?
Yeah, so for the signal transduction, the cell communication lab simulation, Labster, that is individual work. You do have group work this week, and it's to complete your result and discussion final draft from osmosis.
Okay, thank you.
But that's it for lab group work this week. Yeah. Good questions. Allie?
Yeah, sorry, I just thought of another one. For the e-portfolio assignment this week, it is limited to a comic strip, right? We don't have like as many options as before.
Yes, this week we do want you to make like the comic strip. So we thought that would be a fun change.
Okay.
Yeah, if you have a really fun idea that is different than a comic strip, I mean, I'm very open to hearing it, but I thought that would be kind of fun for this week.
Okay, thank you.
Yeah. All right. So Curtis, I just want to make sure your hand is up from a previous question, or do you have another? Okay, perfect. Just checking.
Yeah. No, I forgot to unraise it.
You're good. All right. So let's see. Did we get to everything? Let me check. So we talked about lab, we talked about lecture, we talked about the reviews and the checkpoints. We're feeling good. All right. Well, in that case, again, always pay attention to, again, like what I would really recommend paying attention to is just the home page, the recommended schedule where we have the assignments linked, as well as modules. I'll look into the results thing. I swear, Canvas just wants my, I don't know, just hates me sometimes. Okay. All right. So let's go forward. This is just a summary of lab. Again, individually, you'll do the Labster simulations, but as a group, you'll do the final draft results and discussion. You will be meeting as lab groups with your lab instructor this week. The goal is just to give you one-on-one feedback about your results and discussion sections so that you have really robust feedback on your writing, because that's what we want you to learn, scientific writing. And we'll start a new series of experiments and writing assignments next week. We'll be learning about enzymes. All right, I saw a question. Christina?
Sorry, I was just taking a look at the comic strip thing because of what Ali was talking about. We only pick one of the cell communication topics and make it based off that?
Exactly.
Right?
Yep.
Oh, okay. Yeah.
Yep.
Cool. Thank you.
Yeah, good question. Yep, just pick one, and it's anything you want is good.
Is creative ability being taken into account here?
I mean, as much as possible, but this is not art, it's biology. So as long as you get the concept across, I'm good.
So would it be okay to like just use clip art or pictures on the internet, or do we have to draw stuff?
No, you can use clip art.
Okay, cool.
Yeah, you're good. Yeah, I appreciate anybody that has art background. That's totally fine. People are submitting like the most beautiful stuff, and I'm just like, how do you do that? I'm a clip art girl, so you're good. So, all right. So let's move forward. You guys are so wonderful. I love how much you talk in class. It gives me life. Thank you. All right. So, good. Oh, this is another thing I wanted to cover before we move forward. So this is something that I have been asked a lot, and I thought that it would be a really good time to talk about this. So obviously, we cover a lot of material in this class. We've gone through our unit 1, we're finishing unit 2. We do have like 6 units of content. As we get more and more, obviously, like a lot of this stuff is additive. So hopefully, at this point you understand more about lipids, because we've talked about why they're important with the cell membrane and with the rough ER and stuff like that. So hopefully, like it is additive, and the stuff that we learned in unit 1 really hasn't gone away, because we're talking about how it's applied in kind of more advanced ways, and that will continue, right? So the next unit is metabolism. So we'll be kind of, again, expanding on macromolecules and talking about how we can use those to fuel a cell. So all of these things are really important. But I realize, like biology, and especially this class, is notorious for there's a lot to learn. And so we try to give you a lot of tools to learn it, to supplement and support your understanding. But I realize it's sort of like, well, the question is like, what do I focus on? Like, where do I focus my attention? And I wanted to address that, because it's an excellent question. And I wanted to bring your attention to, again, and what I wanted to mention is that what you should pay attention to is what we tell you to pay attention to in the lectures specifically. So we create the lectures based on like the objectives for any given unit. So you'll notice at the beginning of the lectures, so this is just my lecture from this week about plant communication. You'll always notice that like on the first slide, we tell you the objective is of this series of lectures, or this lecture is this. Like, this is what we want you to get out of the lectures this week. And then the very last one of the very last slides is always, for every lecture we give, is this focus on slide. And as far as like what to know for this class and like what to study for this class, this is where you find that material, right? The reading assignments are excellent, but they are meant to be supportive and they are meant to kind of supplement your understanding, right? Show you where you're struggling or maybe where you need to focus more your attention. But they sometimes go into more depth and cover kind of more topics than we would in the lectures. The purpose of the lectures is for us to explain things in sort of our language of how to understand it. But again, another really important purpose of the lectures is to say, like, here is all of this information that you've learned in the readings and etc. This is what you need to know, right? This is what's important. So those lectures we make are really, 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 really important. So how I would recommend doing this and how I would recommend using these slides is I would pull up a Word document, or, you know, if you like flashcards, use flashcards, whatever you want to do. But I would tell you, so you see the terms here, and then you see the concepts under that. And what I would do for terms is just list them. And this is, again, you have them for every lecture. Some of them are repeating. Cell communication, and then ligands, receptors, whatever, you know. And then what you should do is you should try to define that term. So this is really important, because if you look up the definition of cell communication in your textbook or on Google or wherever you choose to look, it's going to be nonsensical. You're gonna be like, okay, here's a bunch of words that are strung together that, you know, are overwhelming. What you should do is watch these lectures, do the reading, and then look at this term and say, like, okay, based on what I learned, what does this mean? How can you describe it? Like, how would you describe it? Not how would you describe it to one of your colleagues in here, because we are all scientists, right? But maybe think about, how would you describe this term to, like, for example, my mom has like no background in science. So I always say, like, how can I describe this, or how would I describe this to my mom? So cell communication, like off the top of my head, based on what I know, I would say is how cells communicate with each other, and then how kind of also how cells interpret and like environmental information and process that information to create or to react to that information, right? That's kind of how my mind processes it. It's how cells can communicate, but it's also how cells interpret messages and respond to those messages. So cell communication is sort of like twofold. And then I see ligands. I'm like, okay, again, if you look up ligands on Google or in the textbook, it's going to be something, you know, whatever. To me, it's like a signal that is detected and interpreted and responded to by a cell, right? So that's a ligand. And a receptor is something that detects a ligand or signal. So note, these are not technical definitions. They're not like full sentences. It's how I would describe these things. And this is essential, because if you're describing them, if you're just like rewriting what the textbook says, if you're not paraphrasing it in a way that you understand, you will not remember it. It won't stick, right? I don't even know how cell communication is defined in the textbook, because the way it would be phrased would be like too much, right? It has to be in your own words. It has to be in a way that you understand it. So do that for the terms, and there's terms associated with each lecture. For the concepts, what I would recommend doing is rewriting them as questions. So for example, the first concept is understand how environmental stimuli are detected by cells. I would say, how are environmental stimuli detected by cells? And then again, and try your best to answer it in your own words without looking it up, like without using the textbook or without trying to do this. And then another really important thing is these lectures, these concepts and these terms are taken from the lectures we give. And so you should be able to use the information that was presented in that lecture to answer this. So if I say, how are environmental stimuli detected by cells? Well, in this lecture, we saw that, you know, environmental stimuli serve as ligands that are detected by cellular receptors interpreted by the cell. Oh, that's basic, yeah, and interpreted by the cell. Okay. And that's how I would say that answers that question. So how are environmental stimuli detected by cells? Cells have receptors to detect it, right? So again, very, very important is that I answered that. And I realized that, like, I'm a biologist, but cell signaling is not my field, right? So this is how I would interpret it based on what I learned in these lectures, okay? It's in my own language. It's not from the textbook necessarily, like their words. It's how I would describe this process, okay? How I would describe this process to my mom, okay? Although she would probably tell me what's a ligand. And then I have to describe that too, which is good. So that's what I would recommend doing. I want to mention that as we talked about, like, the information in this class is cumulative. So we are learning more as we go. That we will have a cumulative final, very similar to every checkpoint is cumulative, so that you're constantly studying for the checkpoints, but in doing so, you're studying for the final. But all of these terms and concepts, if you're building like a running study guide like this, you have your study guide for the final, like, created. So do this for every lecture. That is a really good way to study. However you're studying is fine, like what you feel best with. But if I was to tell you, like, what material you should focus your studying on, focus on the stuff that is found on these focus on slides in the lectures. Focus on what we're telling you in the lectures. That is the content that we care about you knowing, right? We obviously want you to know it all, but this is the content that we say, like, well, you gotta know it. So that's what I would recommend doing, kind of asking these questions. You can have this, you can read over it, but if you make these into like flashcards and quiz yourself, that's even better. So that is my best recommendation. I've been asked a couple times if I could make sort of like a guided video on how to do that as well. I'm planning on it. I got to record the lectures for cellular respiration first. It takes a little bit of work, but it's coming. But I just wanted to give that as a preview, since we do have our next unit assessment coming up, that's a really good way that you could study moving forward. So
I have one. Oh, sorry.
no,
I
you're
have
good.
just one question
Yeah.
about the lecture videos. So a lot of the times I find myself more focused on the slide than what is actually being said in the lecture.
Okay.
And then I go to the quiz and I can vaguely remember what, like, if I don't know the question and it's not in my notes, I can vaguely remember that it was being said in the lecture, but I didn't really focus on it. Like, how would you, like, focus, like, I don't know how to explain it, but like, how would you do both? Like, focus on the slide and then focus on what's being said too, like, comprehend it all.
Yeah, it's a lot. I think that one of the reasons we try to keep those lecture videos sort of short, which Jamie is like really good at, and I try really hard. I'm bad at it. I'm not succinct. But, like, you know, I would say that if you were to take notes, like in this class, like, take notes while you're watching those lectures. right? So look at the slide, and yeah, somebody in the chat said like pause the video. Like watch us talk about the slide and then say, what did they say about the slide? And then maybe pause the video and look at what's on the slide. Typically, we try to like keep our information like that we're talking about related to what's on the slide, but sometimes we elaborate with examples, which is sort of like what we try to do. But yes, closed captions are excellent as well. But that's what I would kind of recommend doing is, you know, because they are recorded, you can start and stop them anytime. You're more than welcome to like watch a slide, pause it, look at it, etc. There's lots of really good recommendations in the chat I see coming in too. So
yeah, I'm gonna read those. Thank you.
Yeah, good question. Awesome. Christina?
Something that I thought, I don't know if I should add or not, but like, I find that I think, not trying to be rude or anything, but I find your lectures to be a little bit more helpful because although I love the visuals that Professor Gillen has on her slides, she has like no text on it. So I kind of have to like interpret like the tangent she goes off on for 5 minutes. I mean, she has really good information and it's fascinating and all, but then I have to kind of like figure out what I'm expected to know from all that interesting stuff.
Yeah, I'll kind of mention that to maybe increase the text on the slides too.
Yeah, no offense, because like it's still interesting what she's talking about, and I still understand everything. It's just, yeah, yours seems to be more concise with like what we're expected to know with the text on it.
For sure.
So
I think that
talks so fast. She
does.
talks so fast.
No, she talks fast.
I have this issue where like they were saying pause it and take the notes, you know. Well, I do that sort of to a fault to where you know, her 6-minute lectures turn into half-hour endeavors for me. Which that's the only reason why sometimes I'm like, yeah, it's hers, because it only takes half an hour for me to do the notes instead of a half-hour lecture taking me an hour and 30 minutes. Which is no fault of yours, because again, you sort of slow it down a bit, which is helpful. But yeah, it's like I have to pause like every other sentence just to try and figure out what she said.
Oh no. Yeah, I'll ask her if she can slow down a little bit. It also helps that I'm your professor, so you guys are like, that's her. And also, I'm just like awesome. No, I'm just kidding. Jamie is too. Okay, Christina, is that your
I
hand?
have to put it down. Sorry
okay.
about that.
No, you're good. Jennifer?
Also something that I've noticed, especially like with her videos, is like the questions you make don't seem to like correlate, or I can't correlate them to what she says.
So those questions on the concept checks are actually similar to what you are exposed to through Connect and the reading assignments. So we're trying to tie the content between those sources of information so that there's not a disconnect there.
That makes a lot more sense, because I feel like that was not even remotely covered in the lecture.
Yeah, sometimes we're trying to link the content for you. So that's why we always say like the reading assignments are like before, and then lectures are more like zoning in on exactly what you need to know. Yeah.
I'd also like to say, in case nobody saw my chat, when you're like watching the lectures, with whether it's like Dr. C saying or Professor Gilligan saying it, use the subtitles because they help. Subtitles help me out a lot, especially if I'm writing down like extra details. Like I'll write the notes first, I'll pause it, and then I add like certain like important details. Subtitles, y'all, they help. They really do.
And I hope they're good. We've submitted a bunch of our videos to it's not the Disability Resource Center anymore, it's the SAILS, but so that our captions are not like YouTube generated, that they're like formally generated. They're just, obviously because of everything, it's like very delayed. But they have been submitted. I've also had the request for PowerPoints to be provided instead of just the recorded lectures. So we're going through trying to figure out if we can do that because of like copyright issues is the only reason, but I'm working on it. I want to make it the best that we can and get you guys the content you need. There's a lot of details to it, but I'm working on it. I do appreciate this feedback though. Again, I love that you all talk. It is my favorite thing in the world. Hearing this from you is what I need, right? Feedback is important. So thank you very much for that. Keep it coming. And always feel free to reach out via email too. Like if there's something you need or you would find useful, ask me for it. All right. Okay, cool. So let's move forward. Oh, yes. I have another 30 minutes. Just another reason I love these Tuesday, Thursday classes. All right. So let's get into the content. All right. So, all right. Where was I? Okay. So that's how I'd recommend studying, focusing on the terms, focusing on the concepts in the lectures. The lectures tell you exactly what you need to focus on. The readings are supplemental. Yeah, all right. All right, let's get into some muddy points. Overall, when I was looking through the muddiest points, everybody seemed to have a pretty good understanding of organelle function in general, which made me happy. They're pretty not like super difficult to understand. There were some really good questions about some of the most confusing things. Seem to be sort of like the one big point of confusion was endosymbiosis, not necessarily that people like didn't understand it per se, so much as it's just a challenge to fully understand like what the heck happened. So I thought we could talk a little bit about that. That's one of my passions, obviously, because it has a little to do with microbiology. There were some questions about distinguishing the rough and smooth ER, which I think are excellent questions. And then there was a lot of questions about cell junctions as well. And I think partly when I was reading through, this came to sort of what I think like Jennifer and Christina pointed out, which is there was a little like some discrepancies between sort of what the textbook was asking you to know about these versus what we talked about in the lectures. And again, this comes down to the fact that like the textbook went into excruciating detail, and you don't need to know it in the detail that the text went into. So know the depth for like cell junctions, know the depth that was covered in the lecture. That I think that was like the last lecture. So yeah, and I'll cover that again though, because they can be confusing even then. I do have some topics that I wanted to cover from week 5 that we didn't get to last week, but they are very important, and that was the importance of surface area to volume ratio. And I had somebody ask me a question of, we talked about how like lipids are dissipated by soaps, and this helps to eliminate viruses. So we talked about washing our hands and how that kills viruses. And somebody actually asked me, well then why can't, this was not in our muddiest points, this was outside of this, but I wanted to address it anyway, which was, well then why can't we just drink soap? And it's not just because it doesn't taste bad. So I want to talk about all of these things if we can. All right. So let's get started. So first off, I want to address something, and this is, again, just like in general, a very challenging concept, which is surface area to volume ratio. And I think everybody's sort of understood, like, okay, cells get bigger, okay. But one of the kind of essential things that this comes down to is, why does this matter? So why we talk about in biology surface area to volume ratio is we talk about it because this is the reason that cells are microscopic. And it's because of the fact that as cells get larger, their surface area to volume ratio like decreases, which is not good for cells. And I'll talk about why in a minute. But another reason I want to emphasize this, so actually I was talking to one of my best friends who is Professor Chi-Chi, so some of you have her for lab, but she also is our 182 professor here. So I meet with her quite often. But we were talking yesterday about surface area to volume ratio because we're dorks. But she mentioned, you know, I was like, this is a little hard to explain. And she said, well, it is really important though, because this is also the reason that animals on our planet can't get bigger than they are. And then why certain animals have difficulty regulating processes. So for example, like elephants are humongous. They're very large, but they're restricted by that size. They won't get larger than that because actually like the size they are the largest that they can be in order for them to effectively regulate their body temperature. Because as we've talked about, thermoregulation or regulating internal body temperature has to do with being able to detect heat, but it also has to do with being able to release heat. And elephants are basically like the largest that they could possibly be and still regulate their body temperature. Which again, I don't know anything about elephants. So this is why I talk to her all the time. She's just like a wealth of knowledge about things that are not humans, which is, you know, not my thing. But so why? You know, like, why does this matter? So what I want to tell you about is some concepts in geometry, which again, I'm not a math teacher, but this is very important. And if you think about it, is logical, right? So if you think about geometric shapes, cells tend to be, I mean, we think about them as being kind of circular, right? And that has to do with their membrane and the fact that they don't have a lot of rigid structures. So cells kind of have more of a spherical shape. But what you need to know is that surface area, which is the surface of cells, and if you remember, well, hopefully you remember, that the surface of cells, the most external feature of cells, aside from like modifications, is going to be the cell membrane, which is composed of biological lipids. Some cells will have things outside of the biological membrane, like a cell wall or a glycocalyx or whatever the case may be. But for all cells, ubiquitously, cells interact with their environment through the cell membrane. Hold on just a second. All right. But cells interact with their environment through the cell membrane, right? So if you're looking at this picture, imagine that this is a cell. The gray part of this is the cell membrane, right? Sort of like the skin of the cell. But inside of the cell, there's a cytosol. So the surface of an object is measured in units squared, right? So this is a squared measurement. Inside of that is the volume. So the volume is basically how much of the inside is filled up. But importantly, volumetric measurements are cubic, right? So surface area is squared, volume is cubic measurements. So what this means is that the surface area, like as an object size increases, the surface area will not increase as much as the volume. Okay, so again, as the size of an object increases, it's like a geometric rule due to the fact that surface area is squared and volume is a cubic measurement. As the size of an object increases, its surface area decreases more slowly, or increases more slowly than the volume. So this is really important because inside of a cell is the cytoplasm, and the outside, the surface area of a cell, is the biological membrane. So if you remember, the membrane is how a cell gets nutrients in and gets waste out. So it is basically how it gets all the nutrients to fuel its metabolism and get waste out of the cells. So the membrane is really, really, really important. So if you look down here, this is just comparing potential cell sizes. And you see really tiny, like small, and quite large. But this really tiny cell, like when cells are really tiny, they have a really good surface area to volume ratio. So for example here, the ratio is 3:1. So it has 3 times as much surface area or membrane compared to the amount of cytosol. This means that the cytosol and the metabolism that occurs in that cytosol has tons of membrane to import nutrients, export waste, etc. But what starts to happen is as cells get larger, because again, volume increases in a cubic rate, surface area decreases not as quickly. What starts to happen is as a cell gets larger, this ratio goes down. So you can see small cells 3:1. This, if we increase the cell size by 10, you can see this ratio goes down to like 1/10 of what it was. So 0.3:1. So that means that there's more volume than there is surface area. So there's more metabolic requirements than there is membrane to import nutrients or export waste. And this, again, is emphasized when we increase cell size even more. So what that means is that cell sizes that are large, or cell sizes where the surface area to volume ratio is small, are inefficient to meet cellular needs. So what does this mean? Well, this means one thing. This is a huge rationale for why all cells are small. They have to be small because they have to have way more surface area than they do volume to accommodate the need for nutrient import, waste export. But another thing is that sometimes cells do have to be larger because they have more needs. So cells that secrete stuff, like the cells that make saliva or sweat, right, glandular tissue, or the cells that line like the intestine and have to absorb a lot more nutrients. So they have to move more stuff in or out of the cell. They need more cell membrane. They need a ratio even higher than this. And that's where we get to some of those surface modifications. So one in particular is, I think I kind of talked about it in the lecture, was microvilli. So we talked about the fact that your intestinal epithelial cells have these little extensions of membrane. And the purpose of those microvilli, the purpose of extending the membrane, is if we have more membrane and we have these little surface projections, this increases the surface area of the cell. So again, you should equate, when it comes to cells, you should equate surface area to membrane, because that is the surface of a cell, technically. And so if we have microvilli and we increase the surface area of cell, we have more cell membrane through which we can absorb nutrients or excrete waste or other secretions, right? So that's really what we're talking about with surface area to volume ratio. It's more about why is it important or why does it matter? And it's really comes down to the fact that cells have to be small in order for them to have the appropriate membrane to cytosol ratio or surface area to volume ratio. And if they're larger due to their function, then they're probably going to need some sort of surface modification that kind of increases their surface area. So this is just looking at 2 cells. So this is kind of like a general cell, and this is an intestinal epithelial cell with microvilli. And if you think about it, if you look at my arrow, let's say that my arrow is nutrients that need to get into cells. With this cell, nutrients can pass through the membrane like this, right? All around the cell, whatever. It can absorb a decent amount of nutrients. But if you look at this cell, whose purpose is absorption and has to have a lot of stuff absorbed into the cell, so intestinal epithelial cells are very large. Microvilli mean, again, if you think of my cursor as nutrients, nutrients can not only enter like this, but they also can enter anywhere along the projection of this microvilli. So there's a lot more membrane for way more nutrients to enter and exit a cell. So again, it just kind of explains like surface modifications as necessary. It explains why cells are small. Like I think the largest cell is like an ostrich egg. The largest free living organism is still like, I think it's like this big, which is very, very rare. But it's because like cells and single cellular organisms, because of that, have to be small, because they are basically at the mercy of how much membrane they have to accommodate their cytosol and their metabolic needs. So that's why we talk about surface area to volume ratio. Really, really important, but mainly just important because it just like tells us why, which is a really good question. Okay. Okay. So I want to move on now. I see a question or maybe a comment. Let me see. In the chat, a cell organism the size of an ostrich egg. What kind of, so the organism that is the size, it's the biggest cell is not the size of an ostrich egg. It is smaller than an ostrich egg, but it's like, I can't remember, but I think it was it Zoe or Zara? One of you posted about it in the discussion board a couple of weeks ago. I'll find it. So Christina, I'll find it. But it is a particular organism. It's like the largest single-celled organism, but I think it's about like the size of a quarter, if I remember, which is like the largest thing. Yeah, good question. Okay, so let's talk about one of the, like, again, overall, it seemed like everybody had a pretty good understanding of the organelles. There was 2 organelles that seemed to get a little bit confusing about how to distinguish the 2. And this was the endoplasmic reticulum. So the difference between the rough and the smooth endoplasmic reticulum. So I do want to clarify that the endomembrane system in general encompasses all membrane-bound organelles in a eukaryotic organism. So the endomembrane system is all of the organelles that are composed of membrane. So this would be like the nuclear envelope or the nuclear membrane, the rough ER, the smooth ER, the Golgi, lysosomes, anything that's made of membrane is part of the endomembrane system. So endo means inside, membrane telling you what it's made of. So anything that's inside and made of membrane. On that concept check, one of the questions was which of these is not part of the endomembrane system? And the answer was nucleolus, because the nucleus is bound by a membrane, the nuclear envelope or the nuclear membrane, but the nucleolus is inside the nucleus, and it's actually not composed of membrane. So the nucleolus is not part of the endomembrane system. It's like within the nucleus, but it's not made of membrane. So the purpose collectively, as we will learn more about as we go on, of the nucleus, the endoplasmic reticulum, and the Golgi is essentially to make macromolecules. Primarily, what we're going to be making is basically like protein and lipid. So if you remember from the lectures, ribosomes make protein, right? They take RNA, mRNA that's created in the nucleus, and they take that RNA and turn it into proteins. So we haven't talked about transcription and translation yet. We'll get there in a couple weeks. But basically, ribosomes take mRNA that's made in the nucleus and turn it into protein. So there are 2 types of ribosomes, right? We have bound ribosomes and free ribosomes. So I think I'll go here. The big difference, and again, we will talk more about this later, but the big difference is that when we talk about bound ribosomes, these ribosomes are bound, like what are they bound to? Like binding. They are bound to the rough endoplasmic reticulum. So the rough endoplasmic reticulum is actually named the rough endoplasmic reticulum because of how it looks under a high-powered microscope. So this image is of a scanning electron micrograph of a rough endoplasmic reticulum. And you can see the membrane, the biological membrane of the rough endoplasmic reticulum. And on it, you see these little brown spots. Those are ribosomes. So if you look at that picture and you think about like rubbing it with your fingers like this, it looks, because of the ribosomes, it looks like it would be rough. Like it looks dotted, okay? And I think you can kind of see that here. The rough endoplasmic reticulum, all the ribosomes attached, it looks bumpy, it looks rough. So again, ribosomes make proteins, and so this is just looking more closely at the rough ER. Those ribosomes are attached to the rough ER, and what happens is this is mRNA created in the nucleus. It associates with the ribosomes on the rough ER, and it actually makes proteins, and as it makes them, those proteins enter the rough ER. So you see, again, we have mRNA coming from the nucleus, this ribosome that's bound to the rough ER, making the rough ER rough, creates that protein enters the rough ER, which remember, the ER and the whole endomembrane system in general is composed of biological membrane. And this is where, like, one of the reasons vesicular transport is so important. Because what we can do is if we create a protein and that protein enters the rough endoplasmic reticulum, it can have some stuff happen to it, but don't worry about that. But you'll notice is that protein is folded and then it can exit the rough ER in a vesicle. So this is a vesicle. And remember, the importance of vesicles is vesicular transport. So if we have a protein in a vesicle, because that vesicle is composed of membrane, now that vesicle can fuse with any other cell membrane. It can fuse with another organelle so that this protein becomes part of an organelle. It can fuse with the plasma membrane or the cell membrane so that it can leave a cell. So this would be how secreted proteins, this would be how proteins leave cells. It can also, because that vesicle can fuse with the cell or plasma membrane, that protein can become a part of the cell membrane. So the rough ER helps to create proteins. Most specifically, it helps to create proteins that either leave the cell or become a part of the membrane, because these ribosomes that make the rough ER rough make proteins that end up wrapped in the membrane from the ER. So we'll talk about this more when we talk about transcription and translation. But this is very, very important because you have two types of ribosomes. You have ribosomes that are called free ribosomes that hang out in the cytoplasm. I don't know if I have a picture of that. They hang out with the cytoplasm. Because free ribosomes are making proteins that are not associated with a membrane, those proteins have to stay in the cell. They stay in the cytoplasm. Bound ribosomes make proteins that enter into the rough ER and therefore end up wrapped in a membrane. And so those proteins will like either become a part of a membrane or exit out of the cell. So the rough ER is called the rough ER because of those bound ribosomes. Ribosomes make protein, all right? So you should think rough ER, protein. So rough is referring to texturally how it looks. I mentioned that because smooth is named for the same reason. Like, why are they named rough and smooth? Rough is named rough because of those ribosomes making it look rough. The smooth ER is named for the same reason, which is it's like texture. So the smooth ER, just like you can see in this picture, like looks smooth, but why do we like call it smooth? The smooth ER helps in the synthesis or the creation of specifically lipids. And if you remember, lipids as a macromolecular group, lipids are characterized by being nonpolar or greasy, right? So lipids, think of fats, think of oils. So if you think about like rubbing butter between your fingers or olive oil between your fingers or like whatever lipid you're talking about, it feels greasy, it feels smooth. That's why we call the smooth ER the smooth ER. It creates lipids, but in doing so, it has a lot of nonpolar stuff. It feels like, I guess if you could feel the smooth ER, it would feel smooth compared to the rough ER, which would feel rough because of those ribosomes. So rough ER is rough because of ribosomes. Rough ER makes proteins. Smooth ER is smooth because of lipids. It makes lipids. Lipids are smooth, oil, butter, etc. Importantly, the rough ER does have some other functions. Aside from just making lipids, it also can help in some metabolism. So like for example, in the liver, your liver tends to have a lot of smooth ER because it helps to detoxify drugs, alcohols, and toxins, especially the hydrophobic ones or the nonpolar ones. As well as that, muscle cells are extremely, extremely, extremely high in smooth ER because in muscle cells, the smooth ER helps store calcium ions. And calcium ions are really important for the electrochemical gradients, which fuel muscle contractions. So smooth ER, yes, really important for lipids. Smooth ER is also really important for like detoxification of drugs. So it's like very prevalent in the liver for that reason. Okay. So hopefully that clarifies like rough and smooth. Again, in your mind, just think of like texturally, and that should sort of like signify smooth ER for sure, right? Smooth is smooth because of lipids. And then rough ER is rough because of those ribosomes, and ribosomes make protein, right? Ribosomes and protein. You have to memorize that. But we will talk more about that later on. Okay. So we only have about 5 more minutes, and I do want to make sure that we have plenty of time to talk about endosymbiosis and some other things, which is actually good because this week is different. I mean, lab obligations this week are not nearly as, you don't need to do a ton of group work for lab, so I think like your time in lab is sufficient. So what I want to do on Thursday is meet for lecture and talk about lecture stuff. I want you to meet with me on Thursday. So if you can come to lecture on Thursday, come. We will finish this muddiest points lecture. We can talk about more of whatever you want. Of course, you know, your time is your time. So if you feel like on Thursday you would like to use that time for other class related, you know, whatever, that's fine. I'll be recording it. So I don't record attendance. So you coming to lecture is very, very much appreciated. I think it helps tremendously. But you know, your time is your time. So use it how you wish. I would like you to meet with me for lecture, but if you want to instead meet with your lab groups to work on your results or your discussion, or use that time to do your lab simulation, you're welcome to do that. So yeah. But I think that's where we're going to call it for today. And I will continue the muddiest points lecture on Thursday. And I will post this lecture. Sorry, gross. I will post this lecture under modules. You'll be able to find the recording of this lecture under recorded lectures. But also, since it was related to like week 6 muddiest points, I'll also post it in week 6 under muddiest points. Right. So you'll be able to find it in a couple different places. All right. With that, I'll see you soon. See you Thursday.
Thank you, Dr.
Thank you, Professor.
Thank you. Bye.
Thank
Bye.
you.
Have a
Thank you.
wonderful day.
You too.
You all are so wonderful. Thank you.
Bye. Thank you.
Bye. See ya.
You're even more wonderful.
What is it?
I said you're even more wonderful.
No, you're the most wonderful.
Bye,
And I'm right.
Bye, Dr. C.
Have a good day.
So,
See
Dr.
ya.
C, a quick question.
Yeah.
So on Thursday it will be recorded because I'm just planning on using that for like other class related things, if that's okay.
Totally fine. Yeah, I record. Yeah, I'm gonna record it. So you're good.
Okay. Thank you. Have a great
No
day.
problem. You too. See ya.
Hey, Dr. C.
Hey, how's it going?
It's going okay. I wanted to thank you for they wanted to also say thank you to your donation. It was really kind of you.
Happy to help, but that is a tough situation. It's such a tough time. But
So,
yeah,
thank you so much.
of course. Tell them good luck, and I'll try to help again if I can. Bills, but like that's what I could have. I wanted to do more, but that's what I could do right then. But I like the link's still there, and I'll try my best to do it.
They really appreciated it, both of them, and they wanted to say thank you to you.
No problem. Tell them no problem, and to keep with it. And if I can help further, let me know.
Of course, thank you.
Yeah. I'll hang out for a little bit longer if anybody has questions, but if nobody has questions, all right. Thanks, everybody. All right. Well, I think that I'm probably going to log off then. I do have office hours at noon, so you're more than welcome to use that link if you want to pop over there. But if not, I hope you all have a wonderful day.